everybody. Today I'm going to talk about the structure of the Operon in prokaryotes, and I'll explain how an inducible or repressible Operon regulates gene expression in prokaryotes. And at last, I'll explain how the role of repressors in the regulation of gene expression in prokaryotes. So first, let's take a closer look at these two pictures. An operon is a cluster of functionally related genes that are controlled by a shared operator. Operons consist of multiple genes grouped together with a promoter and an operator. Operons are present in prokaryotes but absent in eukaryotes. In some situations, multiple operons are controlled by the same regulatory protein. In these cases, the operon form a regolen. And an operon is a complete package for gene expression and synthesis of polypeptides. By combining the related genes, all polypeptides required for a specific function that are synthesized in response to a single stimulus. The lag operon is the classic operon example and is responsible for the degradation of the milk protein lactose. The lag operon is an inducible operon. In the absence of lactose, the operator is blocked by a repressor protein. The operon is made up of a promoter with operator and three genes, LAC-Z, LAC-Y, and LAC-A. These three genes are involved in the breakdown of lactose into its metabolites. And the tripoperon is responsible for synthesis of the amino acid, tryptophan. When it's not available in the environment, the tripoperon is made up of a promoter with an operator and five genes that encode enzyme for tryptophan synthesis. The tripoperon is regulated by the regulatory gene TRPR, a gene that is located at a distance from the tripoperon. The tripoperon is an example of repressible operon unless turned off by a repressor protein. The repressor protein is synthesized by TRPR, while the repressor protein is always present in the cell. It is synthesized in an inactive form. When the core repressor is present, in this case tryptophan, it will bind the repressor protein in an allosteric site. And there are various forms of gene regulation, that is, mechanism for controlling which genes get expressed and at what levels. However, a lot of gene regulation occurs at level of transcription. So bacteria have specific regulatory molecules that control whether a particular gene will be transcribed into mRNA. Often, these molecules act by binding to DNA near the gene and helping or blocking the transcription enzyme, the RNA polymerase. So let's take a closer look at how genes are regulated in bacteria. So this first picture, in this bacteria, related genes are often found in a cluster on the chromosome, where they are transcribed from one promoter as a single unit, such as a cluster of genes under control of a single promoter is known as an operon. Operons are common in bacteria, but they're rare in eukaryotes such as humans. Operons aren't just made up of the coding sequence of genes. Instead, they also contain regulatory DNA sequence that control transcription of the operon. Typically, these sequences are binding sites for regulatory proteins, which control how much the operon is transcribed. The promoter or site where RNA polymerase binds is one example of regulatory DNA sequence, and most of the operons have other regulatory DNA sequence in addition to the promoter. These sequences are binding sites for regulatory proteins that turn expression of the operon up or down. And third, some regulatory proteins are repressors that bind to piece of DNA called operators. When bound to this operator, a repressor reduces transcription. And some regulatory proteins are activators. When an activator is bound to its DNA binding site, it will increase the transcription of the operon by helping RNA polymerase bind to the promoter. And at last, many regulatory proteins can themselves be turned on or off by specific small molecules. The small molecule binds to the operon, changing its shape and altering its ability to bind DNA. For instance, an activator may only become active when it's attached to a certain small molecule. And next, I'll explain how the role of repressor in the regulation of gene expression in prokaryotes. So first, in prokaryotic cells, bacteria such as E. coli need amino acids to survive and are able to synthesize many of them. Tryptophan is one such amino acid that E. coli can either ingest from the environment or synthesize using enzymes that are encoded by five genes. These five genes are next to each other in what is called the tryptophan operon. The genes are transcribed into a single mRNA, which is then translated to produce all five enzymes. If tryptophan is present in the environment, then E. coli does not need to synthesize it and the tryp operon is switched off. However, when tryptophan availability is low, the switch controlling the operon is turned on. The mRNA is transcribed, the enzyme proteins are translated, and tryptophan is synthesized. The five genes that are needed to synthesize tryptophan in E. coli are located next to each other in the tryp operon. When tryptophan is plentiful, two tryptophan molecules bind the repressor protein at their operator sequence. When it's absent, the repressor protein does not bind to the operator and the genes are transcribed. So just as the tryptophan is negatively regulated by tryptophan molecules, there are proteins that bind to the promoter sequence that acts as positive regulators to turn genes on and activate them. For example, when glucose is scarce, E. coli bacteria can turn on other sugar sources for fuel. To do this, the new genes to produce these alternate sugars must be transcribed. When glucose levels drop, cyclic AMP begins to accumulate in the cell. The cyclic AMP molecule is a signaling molecule that is involved in the glucose and energy metabolism in E. coli. Accumulating CAMP binds to the positive regulator catabolite activator protein, a protein that binds to the promoters of operons which controls the processing of alternative sugars. When CAMP binds to CAP, the complex then binds to the promoter region of the genes that are needed to use the alternate sugar source and transcriptional activation by the CAP protein. When glucose levels fall, E. coli may use other sugars to fulfill but must transcribe new genes to do so. So as glucose supplies become limited, CAMP levels increase. This CAMP binds to the CAP protein, a positive regulator that binds to the promoter region upstream of the genes required to use other sugar source. And the third type of gene regulation in prokaryotic cell occurs through inducible operons, which have proteins that binds to activate or repress transcription depending on the local environment and the needs of the cell. The lac operon is a typical inducible operon, as mentioned previously. E. coli is able to use other sugars as energy source when glucose concentrations are low. One such sugar source is lactose. The lac operon encodes the genes necessary to acquire and process the lactose from the local environment. Regulation of the lac operon. The transcription of the lac operon is carefully regulated so that its expression only occurs when glucose is limited and lactose is present to serve as an alternative fuel source. So I hope you guys enjoyed the 3 points that I have to talk about.